Good afternoon, dear friends. In last lecture, I have discussed the different types of microscopy. Today, we will see the next point, that is sterilization. So, what is sterilization? Yes, what is sterilization? It is the process by which the article or the surface of medium is made free of all type of microorganism. It may be either a vegetative or the spore form. Yes, it is the process by which an article or the surface or by which the article or the surface or the medium is made free of all microorganism either in vegetative or the spore form. So sterilization is the process of destruction. Another definition of sterilization is the process of destruction of all form of living microorganism from a substance. We will see the different terms. First, we will sterilization, then second, disinfection. Yes, it is the process of destruction of all pathogens or the all microorganisms capable of producing the infection in living cell, but not necessarily the spores. The all organism may not killed, but the number is reduced at a level that is no longer harmful to the health. That is disinfection. Next term, that is disinfectant. Yes, disinfectants, these are the antimicrobial agents that are applied to the surface of non-living object in order to destroy the microorganisms that are living on the object. So what is disinfectant? These are the microbial agents that are applied to a surface of non-living object to destroy the microorganisms and that are living on the object. Next, that is antiseptic. These are the chemicals which can safely be applied to a living tissue and are used to prevent the infection by inhibiting the growth of microorganism. That is antiseptic. Next, asepsis. It is the technique by which the occurrence of infection into a uninfected tissue is prevented. Next, bactericide or the bactericidal agents or the germicides. So first we will see the bactericidal. These are the chemical substances or these are the agents which able to kill the bacteria. And what is germicide? These are the chemical substance which are able to kill the germs. That is germicidal and bactericidal. Yes, cidal, it means they kill the bacteria, kill, and static, it means prevent the growth. Keep in mind the difference. Cidal. So what is bactericidal? It means they kills the bacteria. And what is bacteriostatic? It only prevent the growth of bacteria. Same here. The what is germicidal? It kills the germs. And what is germistatic? It prevent the growth of germ. Yes, these are the different terms that are used in sterilization. In sterilization, the articles having direct application on human and animals are subjected to sterilization. Then this material, it includes the drugs, nutraceuticals and surgical equipments and also the food. Then sterilization is done to preserve the substance for long time without decay. Yes, sterilization is done to preserve the substances for long time without decay. Then secondly, the substance that is not sterilized, it may contain the microbes which may causes what? Which may causes the infection when it is consumed or when it is administered. Where the substance that is not sterile, it may causes what? Or it may contain microbes and it may causes the infection when it is consumed or when it is administered. Second thing, the microbes are invisible to naked eyes even those like bacteria have a protective sheath on their surface and making them resistant to sterilization. For this effective sterilization techniques for this effective sterilization techniques are designed and studied in microbiology. Next, we will see the classification of sterilization or the methods of sterilization. There are basic or the main three methods of sterilization. First, physical method, then chemical method, and third, mechanical method or the filtration method. Yes, what I am saying, there are main three methods of sterilization. First, physical method of sterilization, second, chemical method of sterilization, and third, mechanical or the filtration method of sterilization. We will see in detail classification. First, in physical method, in that first, sunlight, then dry heat sterilization, moist heat sterilization, and radiation sterilization. Yes, in that first, in physical method, first by use of sunlight, then by use of dry heat sterilization, then moist heat sterilization, and radiation sterilization. In dry heat sterilization, it is again divided into first in incineration, then by use of red heat, then by use of flaming, and last that is hot air oven. In dry heat sterilization, it is again divided into first incineration, then red heat, then flaming, and last that is hot air oven. Next, moist heat sterilization. In that first, by use of autoclave, then tyndalization and pasteurization. In moist sterilization, it is again divided into first autoclave, then second that is tyndalization, and third pasteurization. Then next, radiation sterilization. In that first, by use of ionizing radiation, and second by use of non-ionizing radiation. Yes, first that is by use of ionizing radiation or the ionization radiation, and second by use of non-ionizing radiations. So this is the physical methods. Second, the chemical method of sterilization. First, in that first, sterilization by heating with bactericide. I have discussed. So what is bactericide and what is germicides? First, the sterilization by heating with bactericide, and second that is gaseous sterilization. This is the chemical method. In chemical method, first that is sterilization by heating with bactericide, and second gaseous sterilization. Then third method, that is mechanical or the mechanical or the filtration method. In that first, membrane filters, then seeds filters, then candle filters, then sintered glass filter, and last that is HEPA filter. Yes, in mechanical or the filtration methods of sterilization, first membrane filter, then seeds filter, then candle filter, then sintered glass filter, and HEPA filter. So these are the methods of sterilization: physical method, chemical method, and last that is mechanical or the filtration methods. We will see in detail one by one. First, that is physical method. Yes, this is the most common. First, that is physical method. It is the most common method of sterilization. Then the heat is in this method the heat is used to kill the microbes in the substances. First, what I am saying, this is the most common method of sterilization. Then second, in this method the heat is used to kill the microorganism or the microbes in the substances. Then the temperature of heat and the duration of heat are the factors that affect on the extent of sterilization. Yes, in this method the temperature of heat and the duration of heat are the factors which are affect on the extent of sterilization. Then in heat sterilization, In heat sterilization, it means what? That is physical method. It means in physical method the heat is used. Keep in mind, yes, in physical method of sterilization there is use of heat in order to kill the microbes which are present in the substances. Yes, in heat sterilization process, the longer the exposure to heat is better in the sterilization at a given temperature. As the temperature of heat rises, the time span required for the sterilization is decreases. Yes, what is the proportion? That is, as the temperature of heat is rises, the time span required for sterilization is decreases. Further, the sterilization time is decreases with decrease in temperature. Yes, sterilization time is increases with decrease in temperature and vice versa. Yes, keep in mind what is proportion. The sterilization time is increases with what? With decrease in temperature and the vice versa. But one needs to maintain the minimum sterilization time or a minimum contact time for the heat to touch with microbes or the bacteria, and thereby killing the bacteria or the microbes. Yes, in physical method, first that is by the use of sunlight. Yes, in sunlight first, the sunlight it possesses the germicidal activity due to the combined effect of UV, that is ultraviolet rays and heat rays. The sunlight it possesses the germicidal activity due to the combined effect of the UV rays and the heat rays. Thus, it effectively kills the bacteria in water and other surfaces of earth in tropical countries. The sunlight can even kill the bacilli in 2 hours. Here, the sunlight can even kill the bacilli in 2 hours. However, the effectiveness is depend upon the UV rays which are passing through the ozone atmosphere and the amount of heat it can be produces. Yes, what I am saying, the sunlight can even kill the bacilli in 2 hours, but which is depend upon what? Which is depend upon the amount of UV rays passing through the ozone atmosphere and the amount of heat it can be produced. And this method is clinically not in practice. Keep in mind, this method of sterilization is clinically not in practice. So this is the first, that is sunlight. Second, we will see the dry heat sterilization. In dry heat sterilization, first that is incineration, then red heat, then flaming, and third that is hot airborne. The dry heat sterilization, it is the most reliable method. Second, it is also more widely used sterilization method in which the enzymes and other essential cell constituents are destroyed. The heat sterilization is the most reliable and widely used sterilization method in which the enzymes and other essential cell constituents are destroyed. Then this process is more effective in hydrated state, where the hydrolysis and the denaturation is occur under the humidity conditions and thus requiring the low heat inputs, while in dry state the oxidation occurs and which requires the heat inputs. I am repeating, this process is more effective in hydrated state where the hydrolysis and denaturation occur under the high humidity condition. Then this requires the low heat input, while in dry state there are two states basically. First, that is hydrated state, and second, dry state. In hydrated state, where the hydrolysis and in dry in hydrated state there is occurrence of hydrolysis and denaturation under the high humidity condition, and thus the requiring lower heat input, and thus it requires lower heat input. And in dried state it occurs what? There is occurrence of oxidation and which requires high heat input. Yes, there are basic two state, hydrated state and dry state. In hydrated state, there is occurrence of hydrolysis and denaturation under high humidity condition, and therefore it requires the low heat input. And in dried state, there is occurrence of what? Oxidation, therefore which requires the high heat input. Then we will see the principle of dry heat sterilization. The oxidation process is involved in the killing of organism by dry heat sterilization. In this, the dry heat sterilization, the oxidation process is involved in the killing of organism by dry heat sterilization. Then this oxidation process is less effective than the hydrolytic damage. This oxidation process is less effective than the hydrolytic damage, and which involves exposure to steam. Then therefore the dry heat sterilization is usually employs the higher temperature. Therefore the dry heat sterilization is usually employs, or it requires higher temperature, which is ranging from 160 to 180, and time for this 2 hours. Then we will see first, that is incineration procedure. Incineration, in this it is done especially for inoculating loop used in microbe cultures, that is nichrome wire loop. Yes, in incineration it is used for sterilization of inoculating loop, that is nichrome wire loop, which is used in microbe cultures. The metallic end of loop is heated to red hot on the flame. This exposure kills all germs which are present on the inoculating loop. The metallic end of this loop, that is nichrome wire loop, is heated to red hot, heated up to the or until it become red hot on the flame, and this exposure will kill the germ. Next, that is red heat. In this procedure, the articles are exposed to flame till they become a red hot. In this procedure, the article it means the substance or those objects which are going to be sterile, or which want to be sterile. It means in this procedure the articles are subjected to a flame till they become red hot. It is used for sterilizing the tips and points of spatula. It is used for sterilizing the tips and the points of spatula, forceps, then inoculating loop, knives, etc. The points of forceps, inoculating loop, inoculating wire and loop are sterilized by heating them almost vertically in a Bunsen flame till they become a red hot. Then next method, that is flaming. So this is the process of exposing the metallic device like the needle, scalpel and scissor to flame for a few minutes. This fire burns the microbes and other dust on the instrument directly. So this is the incineration, red hot and flaming method. Next we will see the hot air oven. As I said this, or as I discussed in earlier practical, that is hot air oven, it is dry heat sterilization. Dry heat sterilization, it means the sterilization by the use of hot air oven. Yes, hot air oven. This method is suitable for dry material, for example, or the like powders, metals, then devices and glasswares. This method is suitable for dry materials, for example, powder, metal, or metal devices, or that is metallic devices or the glasswares. Here the thermostable material on a racks inside the hot air oven. Then in the closed oven, hot air is circulated at particular temperature and time. Next we will see the construction and principle of this hot air oven. The hot air oven consists of a double walled chamber of aluminium or the stainless steel. Yes, first point, this hot air oven, it consists of double walled chamber of what? Aluminium or the stainless steel, and which are separated stainless steel and separated from the outer case by a thick layer of insulation made of fiberglass insulation, and is also filled in the allow flag door, and which carries the asbestos jacket that provides the tight seal. Then heating is affected by electrical heating element, and the thermostat automatically control temperature. The heating is affected by electrical heating element and thermostatic knob. It is which automatically control the temperature, that is temperature controlling knob. An oven is based on the principle where the sterilization is accomplished by dry or the hot air is less effective as compared to moist heat sterilization. Is less effective than the moist heat sterilization because in presence of moisture the proteins are easily coagulated, and moist heat sterilization has greater penetrating power than the dry heat sterilization. Yes, by the process of protein denaturation and the oxidation, the microbes are destroyed by dry heat for normal sterilization. Yes, by the process of what? First, protein denaturation and the oxidation, the microbes are destroyed by dry heat sterilization. For normal sterilization, it should be operated at 160 degrees Celsius for 2 hours. Yes, next we will see the temperature time relationship. Next we will see the temperature time relationship for hot air oven. Yes, for temperature 140, time that is 3 hours. Then temperature 150, for that time it is 2.5 hours. Then for 160 degrees Celsius temperature, for 160 degrees Celsius temperature time is 2 hours. Then 170 degrees Celsius time is 1 hour. Then 180 degrees Celsius time that is zero point 0.5 hours, it means half hour. This is the temperature time relationship. This is important for MCQ, the temperature time relationship for hot air oven. Then we will see the merits of this hot air oven. The installation and operation cost for dry sterilization is less. Then heat can easily penetrate the material. Then the dry heat sterilization is non-toxic and does not harm to the environment. Then next merit, it is non-corrosive to metals and sharp instrument. These are the merits. Then we will see the demerits. It is time consuming method due to the slow rate of heat penetration and the microbial killing. Next, demerit, most of the material can't without stand the most of the material can't withstand a high temperature. So this is the merits and demerits of hot air oven. Next we will see the application. Hot air oven are used to sterilize the glasswares, forceps, then scissors, scalpels, or glass syringes, then cotton swab, petri dishes, test tubes, and pipettes. Then the hot air oven also is used for sterilization of fixed oil, Then powders, for example zinc oxide powder, talcum powder. Then also used for sterilization of glycerin, liquid paraffin, then sterilization of propylene glycol, then waxes, and the other particles that are either spoiled or not effectively sterilized by the moist heat sterilization. So these are the applications of dry heat sterilization. But one note for this: first, one note, that is, first over must be allowed to cool for about 1 hour before opening the doors, otherwise glasswares are likely to get cracked. Second, it should not be overloaded. Then third note, do not keep the material which will get decomposed at do not keep the material which will get decomposed at operational temperature. And next, the complete wrapping of article is essential in order to avoid the contamination by non-sterile air. The complete wrapping of the article is essential as in order to avoid the contamination by non-sterile air, which is due to the contraction of the air on the cooling enters the over, and to avoid the subsequent contamination prior to its use. So this is the note for this hot air on. It means the care should be taken, or these points to be considered your during use of hot air oven. This is the use of hot air oven. बोला